Hello everyone, my name is Wise Gulu Jurif, and today we will be talking about the Baltimore Classification system. Just like animals, each virus has its own unique species and classification. As such, viruses are separated into different classifications underneath the Baltimore system. It's a fairly new system in the biology world, only being 50 years old. So first off, we have Class 1 viruses. These are double-strand DNA viruses. These guys are big. This is mostly due to them having a double strand of DNA, which means they need more room to pack all of their genome. So most class 1 viruses replicate in the nucleus. They need to be translated into RNA first before being replicated. Their genomes usually have the origins of replication, which are oocytes. I hope I'm pronouncing that right. Correct me in the comments if I'm not. The oocyte is the place where DNA replication begins, enabling a plasmid to reproduce itself as it must to survive within cells. They use viral or host helicase and primase, and this is due to them needing to convert the DNA into RNA. So small class 1 viruses use host DNA polymers, while the larger ones use their own. The larger ones can pack their own polymers into their capsids, and this is because they are bigger in size. Then we have Class 2 viruses. These are single-strand DNA viruses that have positive or negative strands. This just means how the genome is coded. Positive means they can be directly translated, while negative sense means they have to be converted to positive sense before being translated. Think of negative as backwards, that has to be converted to forwards. I can explain further in a future video. Anyways, Class 2 viruses always replicate in the nucleus. They will convert their genome to complementary strand using viral polymers, then back to the genome strand. They are also really rare in humans, usually infecting animals. Fun fact, parvoviruses are the only family of Class 2 viruses that can infect humans and are also the smallest viruses known. Next up, we have the Class 3 viruses. These are double-strand RNA viruses. They also replicate in the cytosome, and they look like long cellular mRNA strands. The virus codes for its own RNA polymers to create a negative sense RNA that is converted back to positive sense RNA. This is just to help increase the number of positive sense RNA to help with replication. Now we are at Class 5. These RNA viruses are negative sense, and like most RNA viruses, they replicate in the cytosol. The genome looks like long negative sense mRNA. These viruses have to package their own RNA polymer so that it can be converted to a positive sense genome, which then gets converted back to negative sense RNA. Class 6 are also known as retroviruses. These are the popular ones that you most often think of when you hear retroviruses, like HIV. These are positive sense RNA viruses. They replicate in the nucleus where they are integrated into the host genome. They have to package a reverse transcriptase enzyme that converts the RNA into double-strand DNA pretty much right when it enters. Then it gets transported to the nucleus, where it's then integrated into the host genome. After that, it acts like a Class 1 DNA virus when it comes to transcription. The other class of retrovirus is Class 7. These are double-strand DNA viruses. They also replicate in the nucleus. They also have a reverse transcriptase enzyme in their capsid. Double-strand DNA genome goes to the nucleus and as a Class 1 DNA virus to express positive viral mRNA. Then a full-length positive sense RNA is used as a template for the viral reverse transcriptase to make double-strand DNA. And there you have it. That is the Baltimore class system. I can go into a deeper dive into the specific classes and different viruses in them if you want. Just let me know in the comments. Have a good one, folks.